Hello, we are delighted to share our iMeta review, the gut microbiome organ. On behalf of my co-authors across Zhejiang University, Imperial College London, and our international collaborators, I'll take the next 5 minutes to walk you through a single, unifying idea, that the gut microbiome behaves less like a bag of bugs and more like a distributed organ. For decades we described the microbiome by who is there, its taxonomy. But cataloging species tells us little about what the community actually does. So we propose a shift in perspective, the gut microbiome organ. Like any organ, it has a defined spatial architecture, a vast metabolic capacity, talks continuously, in both directions, with the host. Through bioactive metabolites that act like hormones, immune signals, and neuromodulators, it helps set systemic homeostasis and disease risk. 4 messages anchor the review. First, the microbiome can be treated as an organ-like functional system. Second, its metabolites act as endocrine-like mediators linking metabolism, immunity, and the neuroendocrine system. Third, multi-omics and spatial tools are now revealing its functional modules. And fourth, targeting microbiome function, not single species, is the rational path to precision therapeutics. Let's build the organ. Figure 1 shows its anatomy. Communities are arranged along the gut's length and across its radius, from lumen to epithelium. Mucus, epithelial niches, cell antimicrobials, and IgA all enforce host control, and this architecture assembles in early life and is remodeled through aging. Figure 2 is the organ's metabolic engine, 5 interconnected modules: carbohydrate fermentation, amino acid metabolism, bile acid transformation, the TMA and TMAO pathway, and xenobiotic metabolism, that convert what we eat, and what we make, into bioactive signals. Figure 3 zooms into drug metabolism. Microbial enzymes can detoxify a drug, or activate it into something reactive, and they reshape host enzymes too. This is a major, underappreciated source of person-to-person variability in drug response. Figure 4 captures the headline concept: the microbiome as a metabolic, endocrine-like organ. Its metabolites reach distant tissues and engage GPCRs, nuclear receptors, and AHR to tune energy balance, glucose and lipid handling, and cardiovascular function. When this signaling is disrupted, see obesity, type 2 diabetes, fatty liver, and cardiovascular disease. Immunity is the next layer. Figure 5 shows microbial metabolites and molecular patterns being read by host sensors, TLRs, NLRs, and cytosolic sensors, to program immune cell fate, cytokines, and barrier integrity. Lose that dialogue, and you tip toward inflammatory, autoimmune, and even oncological disease. Figure 6 extends the reach to the brain. Through neural, immune, and metabolic routes, the organ shapes the blood-brain barrier and neuroimmune function, linking dysbiosis to conditions from autism to Parkinson's disease. What shapes the organ itself? Figure 7, host genetics, diet and lifestyle, circadian rhythm, and environmental exposures together determine its functional output, and our individual disease risk. How do we read it? Figure 8, integrating metagenomics, metatranscriptomics, metaproteomics, and metabolomics, with spatial and single-cell methods, lets us move from who's there to what they're doing, and to predict disease and therapy response. Figure 9 unifies disease. 3 convergent failures, a breached barrier, metabolic imbalance, and immune miscommunication, form feed-forward loops that connect microbiome dysfunction to disease across nearly every organ system. And we can intervene. Figure 10 lays therapies on a gradient of increasing precision, from FMT and diet, through probiotics and postbiotics, to phage therapy, CRISPR editing, and engineered live biotherapeutics, guided by multi-omics personalization. Figure 11 is our translational compass, an iterative loop from human association, to mechanism, to causal validation in notobiotic models, and back to the clinic, building causal evidence beyond correlation. So, to conclude, the organ framework unifies structure, metabolism, and signaling. It runs on interconnected metabolic, immune, and neuroendocrine circuits, and targeting those circuits, not individual taxa, can move precision microbiome medicine forward.